Hello, students. All right, today is lab day, and we will be performing our lab in a modified fashion since we are not here together. But I do want to explain how the lab actually works. So, we are going to talk about our bacteria lab, which is found on page 82. 82. And here's the deal. We're going to fill this out and we're going to do this together. And I have collected some samples and I'm going to show you the results. All right. So the problem of today's lab is what location in the school supports the greatest population of bacteria and why? We're going to go on a mission. We're going to go on a hunt and try and find a location that has the greatest number of bacteria. Now, normally I would have you pair up and in your lab group go around the school and collect samples using a sterile swab like this one here. And when you bring it back to the lab, just rolling it gently on the auger in the petri dish. Then we would close up the petri dish, tape it up, put it into the incubator, wait 3 days, and then open the incubator, but don't open the petri dish, and examine the petri dishes with our unaided eye and then on the stereo microscope in order to get a close-up view of the various little organisms that we collected from around the building. Now, obviously, we're in a modified situation here currently, and so I am going to give you 4 locations here. So in your lab, I would like you to consider the 4 following places: the toilet seat, the bathroom sink, the computer keyboard, or the classroom floor. Which of these 4 locations do you think would support the greatest population of bacteria and why? Don't say it out loud, but take a moment to write your hypothesis. Now, after you've written your hypothesis, you'll see that the procedures are divided into 2 parts, Part A and Part B. Part A taking place on the first day of the week, and Part B being on lab day, which is today. So we're going to pick it up from there. And what I would like you to do before we proceed is in your observation section, you will see the location of samples. We're not using that whole list of 8 different locations. We're just using the first 4. And if you could just write down these 4 locations there and change the numbers to letters. So sample A, toilet seat. Sample B, and we're talking underneath the toilet seat, by the way. Okay. Sample B, the bathroom sink, all around in the sink, just swabbing the whole thing down. Sample C, the keyboard, taking a swab and swabbing the entire keyboard from the computer lab. And sample D from the classroom floor, from your classroom all over the floor, especially around your desks. Okay, these are the 4 locations from which our samples have been taken. And now, again, as I mentioned before, each of those sampling locations were taken with a sterile swab. Sterile meaning clean of all bacteria, no bacteria on these swabs. Swabs brought back and just rolled around on the auger, which you may recall is a nutrient food source for the bacteria to grow and flourish on. We seal up the petri dishes. We put them in a nice, warm, dark location in the incubator. It's dark, warm, and moist. They're upside down so they don't drown in their own waste. And we wait a few days for them to reproduce. Remember, bacteria do reproduce on their own, as long as they have a suitable environment within which to do so. And so we have created the ideal environment, the incubator and the auger, for them to reproduce. And so here's a picture of the incubator in which they have been placed for several days. And there are different shelves in the incubator and a thermometer at the top. There's a door on the incubator that closes so that we can monitor the temperature. And really, the ideal temperature is just below human body temperature. Bacteria seem to love dark, warm, moist areas within which to flourish. So your body is a great incubator for bacteria because we provide all the suitable things that bacteria need in order to flourish. And that could be sad if you are afflicted with a bacterial infection. So, are we ready? I'm going to show you our samples now, and I'm going to show them all to you first quickly, and then I'll go back and show them to you a little bit more slowly so that you can record some observations. Don't worry about writing down what your Petri dish is. We're just looking at the one that you think is the dirtiest. So do you remember those 4 locations? Let me just go back here. Remember, A is the toilet seat, B is the bathroom sink, C is the computer keyboard, and D is the classroom floor. I'm just jotting it down for my reference as well. So let's get to sample A. Here it is. When we look at the Petri dish, look at all of these round spherical structures. These are not individual bacteria, folks. These are colonies. Each one of these round areas is a colony of millions of bacteria. This is what you might expect to see on the bottom of your toilet seat. So next time you sit down to do your business, think about what business the bacteria are up to right beneath your buttocks. They are flourishing in various colonies like this. And again, this is just a view of the colonies of bacteria, not the shape of individual bacteria. So you got a good mental picture of sample A. Let's go now to sample B. Ooh, look at this. This is from the sink, from the bathroom sink. And notice the shapes of these colonies. We've got more rod-shaped colonies going on here, but again, and some spherical too. But remember, these aren't necessarily the shape of individual bacteria. They're just the shape of the colonies themselves. But of sample A and sample B, don't say it out loud, but which one do you think is supporting a greater population of bacteria? The toilet seat or the sink? Well, now let's give you something else to compare it with. C is the computer keyboard. Now, don't pay attention to this geometric shape here. That's just the label on the back of the Petri dish. But let's focus on these 2 fuzzy structures. These 2 fuzzy structures are colonies of bacteria that are flourishing from the keyboards. We could actually draw a smiley face here, and it would almost look like a happy face, wouldn't it? How cool is that? All right, anyway, so here we have 2 flourishing colonies of bacteria, and I'll go back again. Here's sample A, sample B, sample C. Which one do you think is the supporting the greatest population so far? And now we have one more sample to compare it with: the floor in the classroom beneath your feet every day. Check this out. Here is a collection of bacterial cultures from under your feet. From your feet. Think about where you walk each day. Think about what you do with your shoes when you get home every day. Do you wear them through your entire house, or do you take them off at your front door? Think about the appearance of these bacterial colonies as you compare it with samples A, the toilet, B, the sink, C, the keyboard, or D, the floor. So consider those four locations as you pause for a moment to determine which one you think is the nastiest location here. So take a moment and pause. Now, I'm sure you had an idea of which one looked the nastiest, but I have a couple more pictures here to show you, starting with a close-up of that the sample C, that fuzzy stuff that you see here. These are colonies of bacteria growing out in an almost in a heterotrophic format. This is a type of heterotrophic bacteria, and you can review your notes to remember what heterotrophic means. But all of these structures are seeking out new locations for food. That's what that fuzziness appears. Under the microscope, here's what some of our bacteria from the fourth sample up here. And you can see there are some spherical shapes. And what are spherical shaped bacteria called? Cocci. And we have some rod shaped bacteria. What are rod shaped bacteria called? Bacilli. And there's also, although some of them are out of focus, there's some spiral-shaped bacteria around here as well. And spiral-shaped bacteria, what are they called? Spirilli. So we have all three different kinds of bacterial shapes from that fourth and final sample from right under your feet. So think about that as you write the conclusion for your lab, and I have some additional videos for you to watch when you are finished with today's lab. And I just want to remind you to be sure to study for tomorrow's quiz as well. So, on that happy, appetizing note, I am going to bid you adieu by saying bye bye.